What makes proteins the building blocks of life? Welcome to Biochemistry Basics Chapter 3. Subscribe. Discover how proteins are built, purified, and sequenced, the secrets behind modern biology.
When we observe the interior of a cell, we find a dense environment driven by massive protein macrostructures constantly folding and shifting. The diversity of these polymers is immense, yet every one of these structures is constructed from the same set of 20 precursor molecules. To understand how biology achieves this architectural complexity, we must deconstruct these 20 monomers from their first chemical principles. We need to categorize their variable side chains and track how they behave when subjected to different pH environments. These are not static, inert building blocks. Every amino acid is amphoteric. It can act as both an acid that donates protons and a base that accepts them. Because of this dual nature, an amino acid's charge and geometry are never fixed. Its behavior within a folded protein is governed by the chemical microenvironment it inhabits. We begin the structural derivation by isolating the center of the molecule, a sonal alpha carbon atom. To this central carbon, we attach a carboxyl group. Next, we add an amino group. We complete the baseline structure by filling the final two bonds with a hydrogen atom and a variable placeholder, the R group. The electron orbitals around this alpha carbon force these four distinct groups into a tetrahedral configuration. Because the alpha carbon bonds to four different groups, the molecule has two possible spatial arrangements. These stereoisomers are non-superimposable mirror images of each other. Ordinary chemical reactions produce an equal mix of both D and L isomers. Biology, however, is selective. The amino acid residues in protein molecules are exclusively L stereoisomers. This stereospecificity is a requirement for life. Stable, repeating protein substructures cannot form without a uniform geometric standard. In this matrix of the 20 common amino acids, the R group is the only variable that differentiates them. If we sort them by their tendency to interact with water at neutral pH, 5 distinct categories emerge. The nonpolar, aliphatic cluster is hydrophobic. Glycine is the simplest of the group. Because its R group is a single hydrogen atom, it is not chiral. Proline has a rigid, cyclic side chain, limiting the flexibility of any polypeptide where it is present. The aromatic group contains side chains that participate in hydrophobic interactions and absorb ultraviolet light. The polar, uncharged group contains functional groups that form hydrogen bonds. Cysteine is a unique case. 2 cysteine residues can be oxidized to form a covalent disulfide bond, creating a bridge between different parts of a protein. Finally, we group the most hydrophobic R groups, those that carry a net positive or negative charge at neutral pH. All of these properties, size, polarity, and overall charge, set the absolute boundary conditions for how a protein will eventually fold. To see how these rules apply in a biological context, we must analyze the molecule in an aqueous solution. The non-ionic form of an amino acid does not exist in significant amounts in water. Instead, it exists as a zwitterion. The proton shifts from the carboxyl group to the amino group, creating a dipolar ion. We can track how this molecule's net charge changes across the pH scale, starting in a highly acidic environment at pH 1.0. At this pH, both groups are fully protonated, giving the molecule a net positive charge of +1. As we add a base, the pH rises, approaching the first critical threshold. At pH 2.34, we reach pKa1, the point where the carboxyl group loses its proton. Continuing the titration to the midpoint between the two pKa values brings us to pH 5.97, the isoelectric point. At this specific pI, glycine exists as a zwitterion with exactly 0 net electrical charge. Driving the pH even higher eventually leads to the second inflection point. At pH 9.60, the amino group loses its proton, resulting in a net charge of -1. This shows that the electrical charge of an amino acid is a highly dynamic variable, entirely dependent on the local pH. The titration data reveals a significant discrepancy regarding the carboxyl group of glycine. Standard acetic acid has a pKa of 4.76. Glycine's carboxyl group, however, has a pK1 of 2.34. This means glycine's carboxyl group is over 100 times more acidic than that of acetic acid. This difference is explained by pKa perturbation. The positively charged alpha-amino group exerts an electrostatic repulsion that pushes the departing proton away from the nearby carboxyl group. Reciprocally, the electronegative oxygen atoms pull electron density away from the amino group, lowering its pKa as well. The ionizing tendency of any functional group depends on its immediate chemical neighbors. Enzymes use this mechanism to drive catalysis. By positioning specific residues within an active site, proteins can perturb local pKa values to trigger impossible chemical reactions. To build a protein, we must link these monomers into a chain. This occurs via a condensation reaction, where the amino group of one molecule performs a nucleophilic attack on the carboxyl group of another. The process expels water and creates a covalent peptide bond. Because this reaction is thermodynamically unfavorable, the cell must expend energy to activate the carboxyl group before the bond can form. As this process repeats, a polypeptide chain is extruded into the cell, where it begins to fold into its functional shape. The stability and function of the final protein are dictated by fundamental rules, the rigid electrostatics and specific R group properties of its 20 building blocks. When a biological cell ruptures, its membrane splits and spills the internal contents into the surrounding fluid. What pours out is a crude extract, a dense, chaotic swarm of microscopic material. Even in a simple E. coli bacterium, that fluid contains over 3,000 distinct, chemically similar proteins, all mixed together in a single microscopic drop. To understand the specific biological function of just one of those proteins, it must be physically separated from the rest of the mixture. Historically, researchers often viewed proteins as ill-defined, amorphous colloids. Deducing a precise chemical sequence for such large, complex molecules was considered nearly impossible. Frederick Sanger changed that in 1953 by mapping every amino acid residue in the hormone insulin. He proved that proteins are not random accumulations of matter, they possess exact fixed structures. This rigid sequence of amino acids is known as a protein's primary structure. This primary sequence dictates the molecule's distinct physical behavior, specifically its mass and its net charge. The entire field of protein purification relies on exploiting these unique physical fingerprints to force a specific protein out of hiding. To see how charge acts as a fingerprint, we have to look down at the atomic level of a single short polypeptide chain. The interior peptide bonds linking the chain together are covalently joined. They do not ionize, and they contribute nothing to the molecule's overall charge. A protein's acid-base behavior is dictated by the free amino and carboxyl groups at its terminal ends, along with the ionizable R groups branching off the main chain. These specific molecular groups react to their environment. Depending on the pH of the surrounding solution, they will readily gain or lose protons. There is an exact specific pH level where a molecule's positive and negative charges perfectly cancel each other out. This equilibrium is called the isoelectric point, or PI. Because every type of protein is built from a different sequence of amino acids, every protein has its own highly specific isoelectric point. This makes the PI a targetable variable. By engineering the pH of our laboratory tools, we can use these electrostatic properties to separate the protein from the mixture. We can now translate the theoretical concept of charge into a physical laboratory technique known as column chromatography. The basic anatomy of the column is simple. It is a glass tube filled with a solid porous matrix called the stationary phase. A buffered solution, known as the mobile phase, continuously flows through it. In cation exchange chromatography, the matrix beads hold a permanent negative charge. The crude extract is introduced into the column. Proteins carrying a net positive charge snap to the beads, halting their progress. Negatively charged proteins are repelled, gliding through and washing out the bottom. For the proteins trapped inside, the strength of their binding is directly proportional to the magnitude of their positive charge. This creates a procedural challenge. How do we safely remove the target proteins stuck to the matrix without destroying them? The solution is the elution phase. The biochemist alters the salt concentration of the mobile fluid. Highly concentrated salt ions flood the matrix, outcompeting the proteins for the binding sites and returning them to the downward flow. By gradually increasing the salt concentration, the most weakly bound proteins wash out first, while the most strongly bound wash out last. This separates them into distinct fractions. By manipulating the electrostatic environment, a massive percentage of unwanted cellular material is stripped away. Charge separation has limits. Out of 3,000 initial proteins, many in the remaining fraction might coincidentally share a similar isoelectric point. To narrow the field further, we exploit the next physical property encoded by the primary sequence: overall molecular mass. Protein sizes vary significantly. A human cytochrome C protein weighs in at just 13,000 Daltons, while vertebrate muscle proteins can contain nearly 27,000 amino acid residues. Size exclusion chromatography separates the remaining biological mixture strictly by this physical bulk. The column is packed with beads engineered with microscopic cavities. Counterintuitively, the heaviest proteins move fastest. Massive proteins bypass the pores, taking a rapid path around the beads. Smaller proteins wander into the cavities, delayed by the internal paths, migrating slower. By sequentially filtering first by charge and then by mass, the millions of original molecules are whittled down to a highly concentrated few. At this stage, the researcher has a refined test tube. But how do they visually verify if it contains one pure protein or several remaining contaminants? They use polyacrylamide gel electrophoresis, an analytical method that applies an electric field to pull proteins through a cross-linked polymer sieve. A protein's native electrophoretic mobility is complex. It is dictated by the ratio of the molecule's natural net charge to its unique shape, known as its frictional coefficient. To use this gel as a scale for measuring molecular weight, the protein's intrinsic charge and specific folded shape must be completely erased. The chemical equalizer is the detergent SDS. It binds to the protein, unfolding the complex three-dimensional structure into a uniform linear rod. The SDS molecules coat the entire length of the chain, applying a massive, uniform negative charge that completely overpowers the original intrinsic charge. With every protein in the sample now possessing the same shape and charge-to-mass ratio, the electric field pulls them through the gel strictly by their weight. To prove the exact identity of a protein, we temporarily destroy the very structural features we use to isolate it. In some scenarios, we map the entire 3,000 protein proteome of a cell at once, rather than focusing on a single isolated fraction. Researchers use two-dimensional electrophoresis. This method combines our two core physical properties into a single high-resolution matrix. The first dimension maps the x-axis through isoelectric focusing. Proteins migrate horizontally until they park at the exact pH matching their pI. The second dimension forms the y-axis. SGS is applied, and the focused proteins are pulled vertically, sorting them by mass. This sequential technique resolves thousands of molecules. It separates protons that share an identical mass but have different charges, or those with similar charges but different weights. By charting both intrinsic variables simultaneously, the chaotic blur of the crude extract is transformed into a legible map of biology. Returning to the physical purification process, there is a mathematical reality to accept. With every column and filtration step, total material is lost. The goal of protein purification is not to maximize total yield. It is an exercise in targeted elimination. To track this objective, biochemists measure specific activity. Specific activity is a ratio, functional units of target enzyme divided by total milligrams of protein. As purification proceeds, total protein in the denominator falls. Because the target enzyme is retained while nonspecific proteins are discarded, the specific activity increases. A pure protein is achieved when the remaining material reaches a maximum constant specific activity, the point where every molecule in the tube is functionally identical. A protein's three-dimensional shape dictates its biological function. But if that same molecule is unspooled into a linear thread of amino acids, it reveals a highly conserved evolutionary ledger. Every living cell carries a precise, sequential record of its own biological history. Historically, extracting that linear sequence from massive, fragile molecules without destroying them in the process was nearly impossible. The analog hardware of early mass spectrometry simply could not handle intact biological macromolecules. Reading the internal ledger of a protein requires us to solve two distinct problems: a physical barrier of measurement and an algorithmic barrier of interpretation. Physically, we have to force large molecules into a gas phase so a spectrometer can measure their exact atomic mass, without the heat shattering the molecule into unreadable static. Mathematically, once we have that sequence, we need a way to reliably score millions of potential amino acid mutations to calculate exact points of evolutionary divergence across species. We will first examine the physics of electrospray ionization tandem mass spectrometry, the precise mechanism that extracts physical sequence data from a liquid sample. Then we will break down the bioinformatics matrices that decode those atomic weights into phylogenetic history. Proteins are quantitative, physical maps of life's history. To read them, we rely on a strict pipeline of high-voltage physics and algorithmic deduction. Mass spectrometry requires ions to be in a gas phase. For decades, the standard method was heat. But applying enough heat to vaporize a macromolecule causes rapid decomposition, destroying the very sequence you are trying to measure. Electrospray ionization bypasses heat entirely. A liquid solution containing the protein is pumped through a high-voltage capillary needle, which violently disperses the fluid into a fine mist of highly charged microdroplets. As the surrounding solvent evaporates in the vacuum chamber, the intact protein is forced into the gas phase. During this transition, it acquires a variable number of protons from the solvent, giving each molecule a distinct positive charge. This chart displays the raw data output, a mass-to-charge spectrum. It generates a family of distinct peaks, representing the ionized protein species as they hit the detector. The spectrometer only measures the mass divided by the charge. We need the total mass. This leaves us with a single algebraic equation containing two unknowns. Because the ionized species are sequential, the distance between any two neighboring peaks represents exactly one added proton. That equates to a difference of exactly 1 in charge and 1 in mass. Using the measured values from just two adjacent peaks, we can algebraically solve for the exact molecular mass. Through this calculation, the mass of the native protein is determined down to a margin of error of 0.01. These isolated peptides are then fired into the collision cell, a vacuum chamber bled with a tiny amount of a noble gas, like argon. The physical impact between the high-speed peptide and the static gas particles fractures the molecule. Due to the physics of the collision, the breaks occur almost exclusively at the peptide bonds, snapping cleanly between individual amino acids. Crucially, the electrical charge from the original peptide is retained on only 1/2 of the newly created fragment, either the amino terminal side or the carboxyl terminal side. The uncharged fragment becomes invisible to the sensors. This spectrum shows the readout from the second mass spectrometer, which measures those newly charged fragments. Because the collision cell breaks each peptide in only one place on average, each successive peak you see here represents a fragment that is exactly one amino acid shorter than the peak before it. The mathematical distance between these two peaks perfectly equals the atomic mass of the specific amino acid that was broken off in the collision. By reading the mass differences from peak to peak across the entire spectrum, the machine literally spells out the sequence of the peptide. This high-impact fracturing and measurement is repeated for every peptide generated by the initial hydrolysis, until the entire overlapping sequence of the larger protein is definitively mapped. Through high-voltage dispersion and vacuum collisions, we have extracted invisible mass measurements and translated them into a readable text string of amino acids. Extracting a primary structure is only the first step. To trace evolutionary history, we have to mathematically compare that extracted sequence against homologous proteins found in entirely different species. Simple one-to-one matching fails. Over millions of years, evolution inserts gaps and makes single residue substitutions, shifting related sequences completely out of perfect alignment. Reconciling this requires a substitution matrix, an algorithm that assigns mathematical scores to specific amino acid mutations based on their chemical probability. This matrix is Blossom 62. Derived by Stephen and Georgia Hennekoff from thousands of highly conserved sequence blocks, it quantifies exactly how likely one amino acid is to be replaced by another over evolutionary time. The diagonal line straight through the center represents identical matches, where the amino acids stayed exactly the same across both species. Notice that rare, chemically unique amino acids like tryptophan and cysteine receive massive positive scores. Because their specific chemical structures are so hard to replace, evolution strongly conserves them. If two sequences share a tryptophan, it is a high-confidence signal of shared ancestry. Conversely, Common amino acids receive much lower scores for an exact match because they appear frequently and are easily swapped by random chance. Moving off the diagonal, the algorithm scores actual mutations. This is a conservative substitution. Aspartate and glutamate are both negatively charged. Swapping them rarely damages the protein's overall function, so the matrix recognizes this as a highly probable evolutionary step, earning a positive score. But if we compare aspartate to phenylalanine, we see a non-conservative substitution. Swapping a negatively charged molecule for a hydrophobic one alters the folding structure and usually destroys the protein. That organism dies. It is an evolutionary dead end, and the matrix assigns it a severe negative penalty. The algorithm evaluates two proteins by sliding sequences past each other, introducing gaps, and summing these matrix values to mathematically calculate the optimal alignment score. The Blossom 62 matrix turns biological intuition into hard arithmetic, quantifying exactly how much evolutionary distance separates two sequences. We can apply these calculated alignment scores to a highly conserved protein, like GrowEL, which assists in protein folding, and run the comparison across multiple domains of life. The alignment score has an inverse relationship to time. A high score indicates two species diverged very recently. A low score with many heavy penalties reveals an ancient evolutionary split. This diagram shows the literal visual representation of that mathematical divergence, a phylogenetic tree. The physical length of the branches connecting the nodes is directly proportional to the calculated number of amino acid substitutions separating one species from its ancestor. Expanding this process to the GrowEL sequences of dozens of bacterial species generates a massive interconnected map of divergence. When we sequence the proteins inside chloroplasts, the organelles that power plants, they plot directly onto this bacterial tree, mapping tightly alongside cyanobacteria, and mathematically proving their ancient origins as independent, free-living bacteria. By isolating the code hidden within just one single protein family, we can accurately map millions of years of speciation. It starts by relying on the physics of electrospray ionization and vacuum collisions to extract exact atomic mass from fragile molecules. Those physical fragments are then fed into bioinformatic matrices, scoring the chemical probability of substitutions to definitively trace the branches of the tree of life. Biological history is a rigorous, mathematically verifiable fossil record, written permanently into the primary structure of every living cell.
Thanks for watching Biochemistry Basics Chapter 3. If you enjoyed this lesson, please like, subscribe, and turn on the notification bell so you never miss our next adventure into the science of life.